Okay, so we're going to start mixing it up a little bit here. And we're going to start looking at some more specific real world examples. So where we're kind of starting on today here is, this is section 2. We're going to be looking at Mendelian genetics. But before that, kind of a little bit of a vote here. I'm a cat guy. I like cats. I have cats. If I were to take an orange cat with blue eyes, and I were to mate it with a black cat with green eyes, what would the babies look like? You saying like black and? Yeah, the fur, yeah. Let's go with the fur. You can go with the eye colors too if you want to. I'm not expecting you guys to know, but like, what could they look like?
Mix.
Could be a mix. There's really 3 options. So this litter of kittens, if I mix this again, we have different fur types, we have different colors, we have different patterns, we have different eye colors. If I were to mix these two, there's 3 primary options. One is they could still kind of stay pure. You have an orange kitten and a black kitten. Their eyes are closed, but you could assume it'd be the same eye color. The other option is you get a split. I've chosen this photo for a very particular reason, right? Look at the eye color. It's split right down the middle as well. It could be something like that. Or is it, going to be a big giant mix as to what you can get? Different eye, they have slightly different eye colors. They have completely different fur coat types. It's just a big scramble, right? So you kind of got 3 different options here. You could have a pure, you could be a split, you could have a mix. Does anyone have a vote for either way? Who votes A? Who votes B? C? C? Thinking maybe C? So the actual answer here is, it actually depends, because coat, it can be passed on by different genders of cats. There's different pattern types. So it's actually not as obvious, but logically what's going to happen is you're going to have a bit of a mix as to how, but that's not always the answer. The actual answer is you need more specifics to be certain. But as a generality, it's going to mix them up a little bit. So this is kind of the root of Mendelian genetics. You don't need all of this. In fact, you don't need to write any of this down. But this is the guy who starts genetics as we know it. His name is Gregor Mendel. It's Mendelian genetics. It is literally named after him. Again, you do not need to write any of this down. But what you do need to know is that this guy is the person who started it all. And he started it looking at pea plants, which I'll show you a picture of right after. But basically, he was a farmer. He was a monk. And he had, a garden, as they did. And one of the things that he did was he noticed that, some of the peas I'm getting are yellow, and some of the peas I'm getting are green. And he asked a simple question. Why? Why is this happening? He is fertilizing the 2 of them, and he's getting both. What's happening? Some of these produce the yellow, and some of these produce the green. And so he began to look at how genes and traits are passed on. So this is what he did. So he took 2 plants, and he started to pollinate them. Plants are the exact same thing, right? Plants need to pollinate as well. You have male and female plants as well. They pass on the genes. So he took a yellow male and a green female, and he wanted to see what would happen. There's other traits, though, of course, as well. So again, you do not need to write this down. You don't need to write this down. But what happened here is, so he took these peas, and he looked at the color, as well as other traits. We're going to look at just the color for now. So if he has a green one, he has a yellow one, what's going to happen when he breeds these? So this generation is called the parent or the P generation. We'll come back to that later. So basically, this is what happens. I want you to pay attention to the terminology here as well. I have two plants, yellow and a green one. I'm going to breed them. And this is what he got. He got a whole bunch of yellow peas. So my question for you is, if we look around the room, everyone here has dark eyes and dark hair. Brown eyes, I think everyone has brown eyes, right? I think so. Brown eyes and brown hair. How come? Most of the population has those. We have something called dominant genes, for one thing. That's a little bit of a teaser. In this case, I can see that, oh, well, all the peas turned out yellow. Logically speaking, this yellow one here must be some type of a dominant pea. Like it must be a dominant gene. So I go from a parent, a P generation, to a first generation, F1. So I go from P to F1. Now, what do you think is going to happen if I go a step further and I now take these children and I breed these together? What's some possible outcomes? Think about what happened with the cats. It could be a mix. It could be that they're all yellow. Could be that they're all green. What's probably logically going to happen is, well, we're probably going to get all yellow, right? Like that's just what happened with the parent generation. This is what actually happened. So first, your first obvious thing here is you're going to look at this and you're going to be, oh, well, there's yellow and green. But there's one other characteristic I'm hoping someone can find. I'll give you a second just to look at it. See if you can figure out the weird thing here. There's less green. There's more yellow than green. So that's exactly it. Yes. So how does this work? He was curious with this. The reason he chose these things is because they're very, very fast to create generations. You can't do the exact same experiment with people because like a generation's like 20 to 30 years, right? So we look at things that can breed quickly: flies, mosquitoes, plants, etc. Even dogs and animals. So what happened here? How does this work? That's what our goal here is to find out. So one thing that is important to note is the terminology here. We have three different terms. We have P, parent, we have F1, the first generation, we have F2, the second generation. And if I were to go even a step further, F3, that'd be the third generation, right? So one thing that's important to know is we're going to be referencing these terms a lot. It is helpful to know what each of these are. So I actually do want you guys to write all this down. It seems unnecessary right now, but it is important. And we will be referring this exact experiment a fair amount. You don't need to worry about the picture that's in your notes. Granted, it's black and white. So one thing to note is that when he did this experiment, he literally took the peas, he counted them all out, and in total, he had over 8,000 peas that he counted. And this 3-to-1 ratio is very, very important. So this is something that is true for not just color, but for a lot of traits. So he looked at a bunch of different traits, and you do not need to write these down. You're not going to be asked, what are the 7 Mendel traits? But he looked at a bunch of things. So he looked at the plants, the colors of them, the flower colors, the seed pods, the seed, the shape, the texture. Is it rough? Is it smooth? Is it big? Is it small? A whole bunch of things. So he spent a lot of time on this. And in every single case, he found that the F2 generation, right, parent, first generation, second generation, had that 3-to-1 ratio. Did not matter which of these traits. It's all 3-to-1. So how does that work? Why is that the case? That's what Mendel genetics is how do we get to this 3-to-1 ratio? How do genes pass on? So we're starting to introduce some more terminology here, and this is going to be something we're going to use a lot for the next 2 weeks. So first off, what we've concluded is that there are 2 forms of traits, and that each is controlled by a factor. Don't need to write that down. But these traits go by something called an allele. So an allele is a type of a gene. So basically, an allele is a type of a gene. So when we have an allele, if we're looking at color, for example, I have one gene that controls color, and it can either be yellow or it can be green. So what we have, like, how come the first generation was all yellow? Every single one was yellow. What happened? Well, what ended up happening there is yellow was the dominant gene. It controls the traits that appear. And recessive traits get masked. And I will explain that more soon. That's a dominant, think yellow. Or recessive, think green. So I'm going back to this picture here. So right now, let's make sense of this. So I have alleles, I have dominant, I have recessive. What the heck is all this? So an allele in this case, you don't need to worry about writing this down. The allele in this case would be color. I'm just looking at the color of it. Now, of these 2, which one of these is the dominant and recessive gene, as we discovered?
Yellow.
Yellow. Yellow is my dominant one, and green is my recessive one. So there's terminologies in order to understand, like, when we're reading something, we have to understand, like, what does it actually mean? So what, how do I say that, oh, this allele's color, and oh, this gene's dominant, and this gene's recessive? So what we do is we just use capitals or non-capitals. So let's say, for example, I'm looking at color. I could say, hey, dominant is capital, recessive is lowercase. That's the most common way of doing it. So what we do actually, when we model inheritance, don't worry about writing stuff yet. When we model inheritance, the dominant allele is capital and the recessive is lowercase. So I will put this into a chart here that is a lot of words. We're going to break it down in common sense the next one here. So you can start writing now. So dominant is capital, recessive is lowercase. Very, very important, because we're going to start introducing stuff where I'm just going to tell you what the genetics are. So if we have 2 of the same alleles for a trait, we call that homozygous. And if you remember, that's the same. So it's pure yellow or pure green. If it's different, it's heterozygous. So it has some yellow and some green in it. The key thing here, though, is that for heterozygous individuals, for mixed peas, when I mix a green pea and a yellow pea, the dominant trait is going to be observed. This is why in that first generation, every single pea was yellow, because they all had a little bit of that yellow gene in it, which takes control. This is why everyone in the room has brown eyes and brown hair. Those are dominant genes. So again, the key things here are, what does the capital mean? What does the lowercase mean? And in heterozygous individuals, ones that have a mix of those traits, the dominant one is observed. Going back to this, again, right now, what I have is we've already established, we know that this is the dominant, and this is the recessive, right? If I breed these 2, every single one of these is yellow, because every single one of these is heterozygous. They have this trait, one dominant for color, one dominant for the allele color, and then one recessive for the allele color. So when I breed these, when I take a yellow pea with this, and I breed them together, I get this. These are the 4 outcomes. Remember that 3-to-1 ratio? So I'm going to get 3 yellow peas to 1 green pea. We've already established that. So this is this is what ends up happening for our F2 generation. Now that worst way of putting it. When we have heterozygous breeding, this is what we get. So I recommend you guys put this in your notes there. Now I gave you those 2 different colors there just so you could do that. When you're done with those, you can please pass those on. But draw this table in there. So actually put this table in your notes. All I need to do now is combine these, right? So what would I be left with in here is I'd get y, it's 2 capitals. Over here, I'd have y and y again. Over here, I'd have a heterozygous. And over here, I'd also have a heterozygous. So what would these all look like? They would all be yellow. But what if I were to breed these? So I'll just give you a second. You don't need to write it down, but I just want you to think. I'm going to give you 30 seconds. What would these end up being? What would you get? Again, if I take this, I split it up. Oops. Think about what type of peas you'll get. I'll give you guys like 20 seconds. Just think about it. So for this first section here, so what will we get here? A yellow or a green pea? It'd be yellow. And we know it's yellow because it's 2 capitals, right? It's definitely dominant. What about on this side over here? Oh, I guess on that side over here. It would still be yellow. Now, it does have a recessive gene, but again, the dominant overrides it, right? But down here, it'd be the exact same thing. I'm still going to get that heterozygous, but on this part, I now am going to get a recessive one. So we're able to use these tables to make a prediction there as to what can happen. Now, if you remember, how many traits was Mendel looking at? 7. It gets more complicated when you add more stuff. We're only going to look at 2 traits at a time. So maybe I'll give you a big green pea and a small green and a small yellow pea. Now I have 2 traits, right? I have size and I have color. We're not going to go deeper than that. We don't need to go deeper than that. We will be equipped to be able to do so, but we don't want to do that. We don't have to do that. So we've already established that again. These peas right here, these are yellow peas, but they carry that recessive gene. We know those are heterozygous, but there's a difference. There's a very clear difference between what something looks like and what its genes and alleles actually are. So we have something called genotype and phenotype to help explain those. Again, you cannot always trust what it looks like, because that's really what this is saying. You can't trust your eyes all the time to accurately predict what it is. You can take 2 black cats, you can make babies, and you might have some orange cats. It can happen. You can take 2 people with brown eyes, they can have a kid, they can have a blue-eyed kid. So When we look at the genes, we look at the genotype. So it's allele pairs, its genes are the genotype, right? Think genes, what's the genetics? Genes, genotype. When we look at what does it look like though? Is this a yellow or a green? We look at the phenotype. So let's get into the nitty-gritty of this. Now, when you pass on genes, usually you pass on 2 alleles. And again, allele is a trait. So think, I don't know, color and size. So basically, again, he looked at all 7 of these like things and he discovered, you know what, there's always 2. There's 2 of these traits that separate during this meiosis thing. Don't need to worry about that. So basically, the big thing here is that 2 of these separate. And when we have a heterozygous organism, think about something like this, big Y, little y, those are called hybrids. Just an important terminology here. Okay, so what we're going to look at here is we're going to start looking at there's going to be a tiny bit of math basically. Like how likely is it if I breed 2 things that I'm going to get if I breed an orange cat and a black cat that I'm going to get an orange cat, for example. So we're going to start looking at stuff like that. So one of these things that we do when we breed hybrids, when we involve hybrids for a single trait, we call that a monohybrid cross. So think about it this way. I'm looking at, I have a pure yellow and a pure green, right? This is a monohybrid cross. Mono means 1. A dihybrid cross, di means two, is when I have two or more of the traits in the same plant. So I think we still have a fair amount to go. We're going to go one level deeper. We're going to start looking at pairs with two alleles and how to predict those. I think we're going to do that tomorrow, though. It's deep. We've gone through so much already. So we got about 20 minutes left. I feel like that's enough time now where you guys have your reading essentials there. This has a lot of those diagrams and stuff, meiosis, something called a Plunkett square, which we'll figure out what that is tomorrow. I figure I'll give you guys those 20 minutes here to try filling in the blanks on those reading essentials. You were given that big, thick booklet there. Go through your textbook. You should have about 10 sections or so that you have to fill out throughout there. But then that'll be it for today, I think. We will finish up Mendelian genetics tomorrow. And then next week, we're going to do an actual experiment with it. You're going to create something that's going to breed with other people's things.